So if we go to lipid absorption, now this is a little bit more complicated. It's like moving a huge couch, like sectional, getting it into the house. Okay, so first of all, two enzymes that break down various lipids are lipase, pancreatic lipase, and phospholipase A. And in terms of the lipid structure, I want to remind you that lipids, most nutritionally important lipids are triglycerides. You can see the triglyceride molecule right here. So triglycerides consist of the molecule of the glycerol part, okay, and fatty acid part. So you have one glycerol molecule and three molecules of free fatty acids. 1, 2, and 3. They're not necessarily identical, okay, that comprise a triglyceride. When lipase facilitates the breakdown of the triglyceride molecule. Question. You go ahead. Okay, when lipase facilitates the breakdown of triglyceride molecule, the fat does not dissociate in glycerol and fatty acids. What happens instead, you have two molecules of free fatty acids and the molecule of so-called monoglyceride, which means one molecule of free fatty acid still hangs on to the triglyceride, to the glycerol. Does that make sense? Right? Make sense? So, phospholipase breaks down phospholipids. Okay, and the products of this breakdown is same fatty acids and the component that is called glycolecithin. Okay. Now, why breaking them down? Now, lipids, how do they behave in the water? They don't dissolve, right? They're hydrophobic. Okay. Now, when you break down lipids, triglycerides, into monoglyceride and free fatty acids, these guys are more hydrophilic and are easier to get dissolved in watery solutions. Not perfect, but better. Okay. So first of all, this breakdown makes them more hydrophilic. Does that make sense? Now we need to step back and take a look at other components of the digestive process, the bile specifically speaking. One of the major components of the bile are the bile salts. They highlighted here some greenish things. The bile salts, what is so interesting about them? What is the they have some interesting properties. They have two groups. They have cholesterol part and they have amino acid part. Now what it makes them? Are they hydrophilic or hydrophobic? Bile salts. Both. They both, okay? They have hydrophobic part, which is cholesterol, and they have hydrophilic part, which is amino acid residue. Right? So what happens? Bile salts, remember we talked about it, they form micelles. Wait, dude, that was too much. Here, they form micelles, where hydrophobic parts turn into the center of the micelle, and hydrophilic are exposed to the outside, to the watery environment of the duodenum. Okay? Now, these micelles accommodate in the center monoglycerides, free fatty acids, and other products of the lipid digestion. Does that make sense? So products of the lipid digestion are packed into the micelles. Okay. Now there's micelles. Okay. So there's droplets of fat coated with the bile salts. They enter by the simple diffusion through the membrane. They enter epithelium. Does that make sense? Okay. Now, what happens in epithelium is quite fascinating. Monoglycerides and free fatty acids recombine together to form triglyceride back again. Remember I told you about sectional. You buy a huge sectional and you try to, and if you want to move it through your front door, you're not going to move it as a sectional. You have to split it in pieces, move each piece separately, but then you're going to reassemble it. Does that make sense? This is what happens here. Triglycerides first are broken into the monoglycerides and free fatty acids, hold into the columnar epithelial cells, and those fragments here, these fragments, are put together back to form triglyceride. Because they're too hydrophobic actually to do anything with them. Okay, they're too hydrophobic, and remember, it's a watery environment in the intestine. In fact, one of the great effects of bile salts you can see here in this picture is that bile salts break big globules of fat into the smaller globules of fat. You can observe it at home. If you have a skillet with oil in it, if you pour water on top of the oil, well, it's going to be in big droplets, big like bunches. If you add dishwasher fluid, or just that hand dishwashing thing, that fat will immediately break down into much smaller pieces. And if you mix it well, you may even see those little micelles. Those are micelles, okay, that fat turns into when you add the dishwashing fluid. And dishwashing fluid is actually a large hydrophobic part and a smaller hydrophilic part. It works just like bile salts. Does that make sense? Okay, so far what we've got, let's take a look again. Okay, let me erase some of this stuff. So we have triglycerides, right, broken into the monoglycerides and free fatty acids. Okay, then we get them packaged in micelles. Then they go into the columnar epithelium. Okay, and then we get them reassembled. Does that make sense? So I have triglycerides, split them into monoglycerides and free fatty acids, package them in the micelles. Micelles get into the columnar epithelium, and monoglycerides and fatty acids, these guys, get reassembled into the triglycerides, into the fat. Now, where fat goes next? First of all, the fluids in the human body, extracellular fluid, lymph, blood, saliva, tears, anything you can think of. What is the major component of those fluids? Water. Not oil, water. Does that make sense? So fats, are they easily dissolved in water? What you got to do with them? You got to transport them on something, right? We have proteins for it. So those fats are attached to the proteins, transport proteins, bless you. And when they are attached to the proteins, they form chylomicrons. Okay? So proteins are easily dissolvable in water-based solutions like blood or lymph, right? When you stick fats to it, they can now carry fats with them. Now, chylomicrons do not enter the blood. In fact, they enter the lymph. And lymph is in the lacteal. I have no idea where the model is, so just trust me. Oh, if you look back, actually, I think Courtney can help us. If you would look towards Courtney, that big model in the center, can you see the whitish vessel in the right villus? Can you show the white vessel? Yeah, in the right villus. The white, yeah, that's the white, you see? That goes all the way up into the villus. Trace it with your finger. Yes, so that white is lacteal. You see that in the center, like a massive one? That's lacteal, that contains lymph. Okay, so that's where chylomicrons go. You may say, wait a minute, but lymph is kind of not a blood. And we have to have it in the blood. Turns out, those lymphatic vessels, they collect lymph. They're like veins. They merge into the bigger and bigger. They merge into the vessels. Vessels merge into ducts. Ducts merge into trunks. And trunks empty into the subclavian veins, right here. Somewhere here. The merge were subclavian and jugular vein. Okay, right here. So eventually, that fat that is in the lymph, okay, those chylomicrons that are in the lymph, they end up in the blood. Does that make sense? And then blood delivers them to the tissues, to the liver, to get, I don't know, deposed into the adipose tissue. Something else can happen to them. Does that make sense? So that just, it doesn't go into the blood, but it not directly. Now, if you would look at the human plasma after the meal, especially fatty meal, the plasma will look cloudy because of the chylomicrons. So you see, yes. Say again. You can think of it as, think about the fat molecule that gets attached to the protein molecule. But you can think of proteins being like an Amazon like box, where you package your fat to deliver it to the tissues, to the liver, to wherever. Does that make sense? That's the function of protein in the chylomicron. Okay. Now you can see, so we have four tubes here in the right bottom corner. 1, 2, 3, and 4. And this is the triglyceride content in each of the samples. And you can see as we progress to the higher triglyceride content, you see how cloudy plasma becomes. And it happens almost immediately. Like if you eat something really fatty, like you have several, I don't know, like a Eclair or some other sweet thing that has a lot of fat, you will have something like that. I don't think so, no. It's mostly carbs. Okay. Does that make sense? So that's how fat is being digested. Now this is summary, and you see summary so far. Okay. I'm going to trip myself. Okay. Not okay. All right. Hopefully that will prevent me from tripping. Carbohydrates. In which two places carbohydrates are digested? Mouth and duodenum. Mouth and duodenum. Okay. In both cases, it's amylase. It's the salivary amylase in in the mouth, right? And pancreatic amylase in the duodenum. And a bunch of smaller guys like brush border enzymes right here that will break down oligosaccharides into the ever so smaller pieces. We cool? Proteins, stomach and small intestine, right? Pancreatic, so pepsin breaks down proteins in the stomach. Trypsin, chymotrypsin, elastase, carboxypeptidase, pancreatic enzymes that break down protein in the duodenum. And brush border enzymes, aminopeptidase and dipeptidase, break down it even further. Right? Does that make sense? Fat is broken down in all three locations: mouth, stomach, and intestines. Right? In the mouth, it's lingual lipase. In the stomach, it's gastric lipase. In the duodenum, it's pancreatic lipase. Right? And then, remember, those products of breakdown, they have to be emulsified. They have to be packaged in the micelles. Emulsification, okay, that sometimes causes. So emulsification of lipids. What's emulsion? No idea? Emulsion, okay, what is the solution? What is solution? What's solution? Like I have tea here. Is that a solution? Yeah, it's a true solution. Where chemicals exist in the form of molecules or ions in the water. So it's water solution of chemicals. That makes sense? Milk, is it a solution? True solution. No. Why? What happens to the good milk, like really good milk, that you leave in the fridge for a couple of days? Not homogenized milk, not the one that you buy in store, but say you buy it from the farm. It doesn't matter if it's pasteurized or not, non-homogenized. Yes, into what? Well, what's on top? The cream, the fat. And on the bottom, it's like a skim part, right? So milk is not a true solution. So-called colloidal solution. Milk is a lot is kind of underestimation of tiny small little fat droplets in the water. Of course, there's a bunch of proteins and stuff. Okay, does that make sense? So when you leave it, the fat starts to slowly separate and goes up because it's lighter than water. If you take this bottle of milk and shake it, it's homogeneous again. So it's colloidal. It's not a true solution. It's colloidal solution. Solution of liquid in liquid, like fat in water, is called emulsion. Liquid in liquid. Okay? That's emulsion. Some drugs are emulsions. If you would look at like liquid drugs, they can be emulsions, especially topical ones. Does that make sense? That's emulsion. They can separate. And you have to shake it, like shake it well before use. It's probably emulsion. Okay? Now, when we say, again, if you will take oil, mix it with water, and shake it. You know those lumps? And there's a blob of something that floats up and down? Like, a what? Yeah. That's whatever blob it is. If you shake it, it's not going to form emulsion. It's probably going to form like smaller blobs and they will merge all together again. Okay? Now, same will happen if you mix water and oil. But if you add a little bit of the dishwasher fluid, you will emulsify the fat. You shake it, and now fat exists in really tiny, small droplets. Does that make sense? Okay, so those bile salts, they emulsify lipids. They break down large lipid globules into the smaller and smaller and smaller droplets. Does that make sense? No, they will not. They will not. I mean, as long as bile salts are present, they won't be able to merge. It's all about surface tension and minimizing the surface tension. Okay? What I wanted to say. So, speaking of, what produces bile? Liver. If we get rid of the liver, what's going to happen to the patient? Yeah, patient's going to die really quickly, so it's not an option. Is there another organ that also kind of relevant to bile? Gallbladder. What does gallbladder do? It stores and concentrates it, right? Stores and concentrates. Okay, if somebody gets gallbladder removed, which type of food this person should eat with caution? Fatty food. Does that make sense? Because when fat will enter the duodenum, there will be no bile ready to emulsify right away. It will take time for liver to produce enough bile salts. Does that make sense? Yes. Question?
So like what happens if they continuously eat like a fatty diet in their gallbladder?
Malabsorption. Malabsorption and so-called steatorrhea. You know what steatorrhea is? When you sheet fat. There's a lot of undigested, unabsorbed fat in the feces. Feces are going to float. Normally feces think. They don't think. Well, in some cases they do. Feces sink in the toilet. Okay? That makes sense? Probably just because recently we watched one of the episodes of Futurama. Oh, steatorrhea? It's not normal. I mean, it's not normal. You can notice it. You can notice it, and usually a malabsorption. I don't. People who eat fatty, well, if they repeatedly eat fat enough to give them steatorrhea, then I assume they will become obese just because it's a lot of fat. Okay? I mean, if they eat carefully a little bit of nuts, it's not going to cause steatorrhea in them. That makes sense? Now, steatorrhea sometimes can be observed after really excessive drinking, when liver kind of gives up for the night and fat accumulates, and in the morning you see what you produce and it's grayish and floats. But it's really excessive drinking. And that will go away when liver will resume its normal function. Okay? Does that make sense? So lack of bile will lead to the malabsorption of fats. But other consequence of it can be the malabsorption of fat-soluble vitamins. So if they can't absorb fats, they cannot absorb vitamin A, vitamin D, vitamin K, vitamin E, and that will create other problems. That makes sense? Nucleic acid, not interesting. I mean, they're not really, how to say, a nutrient in essence, okay? And the percentage of the amount of nucleic acid that we get from food is not very, very large. So they are processed in the small intestine by nucleases. So ribo and deoxyribonucleases, they split molecules of DNA and RNA into the individual nucleotides, and then enzymes of the brush border, nucleosidases and phosphatases, break them further into the nitrogenous bases, phosphates, and sugars. Now, I have a question for you. If they are not energy carrying nutrients, what is the benefit that we can get from them? What can we use pentose sugars, nitrogen bases, and phosphates for? Simple question. DNA and RNA synthesis, we can combine them. Cells can combine them back together and form nucleotides to form DNA and RNA. That make sense? Okay. Now, before we move on to finish our trip through the alimentary tract, why? What? Endoscopy is fun. Colonoscopy is probably even more fun. Not for the one who experiences. So for digestion, that's probably the one of the. So this is the physiology part of the digestive activities. So you have to understand what those enzymes in the duodenum do. Okay, what they function is. What is the substrate? What are the products? How trypsin regulates everything else, activates all other enzymes, and how enterokinase activates trypsin, how nutrients are absorbed. Secondary active transport, micelles, where's what is the role for sodium ion to, yeah, question? Yes, question no. What is the role for sodium ion to maintain the gradient to facilitate the secondary active transport? Of course, the apical membrane, right? And of course, the hormones of duodenum, cholecystokinin and secretin, those two. Cool? Okay, let's move to the rectum. Colon first, but then rectum. Anatomically, when small intestine is over, now 3 parts of small intestine, can anyone name them? Duodenum, jejunum, and ileum. In this order, okay? So ileum is the last stop before you leave small intestine and enter large. And they are separated, small and large intestine, by the ileocecal valve. That makes sense? I'm gonna do the walk through this large intestine, and we will leave through the anus. Through the ileocecal valve, chyme enters the cecum. From cecum, which is a large pouch with only one exit, into the ascending, going up colon, chyme enters the ascending colon. From ascending colon, it goes into the transverse colon. From transverse into the descending colon, then to the sigmoid. Sigmoid, name comes from the Greek letter sigma, okay? Shaped, bent part. And then from sigmoid colon, it enters the rectum. And it's not chyme anymore, it's feces. So feces leave rectum via anus. Does that make sense? Now, step by step. Well, cecum, okay? Here, absorption of water and electrolytes. Cecum has a lot of bacteria. Your small intestine is surprisingly clean. There are very few microbes in the small intestine. Most of your digestive microbes are in the large one. And most of them are in cecum. I forgot to ask my wife to send me the pictures of mice. So she works with the, I can try and call her during the break, and she can email them to me maybe by the end of the class. Now, she works with mice that completely lack digestive microbes. They're sterile. They have no microbes in the intestines whatsoever. Their feces don't smell. I'm not kidding. They don't smell. Okay? Huh? It's going to get microbes right away. You got to keep them sterile. And it's not an easy task. Really, it's, to be honest, it's a pain in the ass. It's a lot of physical work. Because you have, now think about this. One thing is operate something like plates, microbiology plates on a sterile level. Another thing is to operate cages in a complete isolation, in an isolation chamber, wearing gigantic rubber gloves, being completely gowned. It's really kind of a tough physical job. Now, interestingly enough, if you would compare cecum of the mice that are germ-free, that do not have any microbes, and mice that do have microbes, mice without microbes have ginormous cecums. As my wife described it, she came to the animal facility and she opened the cage, and the first thought was, those are males, they shouldn't be pregnant. Those were males, but they looked like they were pregnant. It was really gigantic. Like we talk about mouse. Mouse is like this big. It was like 2-3 inches in size. It's huge. Cecum can be huge. So that those microbes are important, really important. And they assist us in digestion. You like sushi, right? Well, at least some of you like sushi. You don't like sushi. Okay, I do like sushi. Do you like sushi? Well, sushi, one of the components there is the seaweed. Turns out we have special microbe in the guts that digests seaweed. We can't digest seaweed as humans without microbe. That particular, I don't remember the species. Okay, so that's why we can't eat sushi.
So is that why like koalas have like super long cecums because of all their plants?
Probably because of the diet, but it's like cows have a different digestive system because of what they eat. Koalas probably too have different structure. Maybe they have less microbes, or maybe they have more microbes, or maybe they have different microbes. Okay, but that actually the microbiome can, as you can say, can have anatomical impact. Okay, change anatomy of the large intestine. Here's a little attachment here to the cecum called vermiform appendix. For a long time, we didn't know what the function is. Honestly, we don't know what the function is now. We're not sure, you know. Like, we know what the function of the stomach is. We know, you know, it digests. With appendix, it's more complicated because, first of all, it can get inflamed. We all know the disease is called appendicitis, and everybody freaks out about appendicitis. Why? If it bursts, what happens? All microbes that are in the appendix are going to spill into the peritoneal cavity. Now it's called peritonitis. And those microbes are anaerobic. They will have a field day. No oxygen, they will just grow. So in some cases, one of the first treatments, open up and expose them to the air. It will control the growth. So just open up peritoneal cavity and expose the contaminated guts to the air. So microbes will start to die. Hyperbaric chamber also helps. Anyway, appendix can inflame, and it's really very unpleasant, I suppose. Never had one. Yes, not appendix, but appendix. Yeah, I'm getting there. So for some time, appendix, there was an idea, let's just remove appendix from everyone, just deliberately, you know. Let's get it out. Apparently, it doesn't do much. Now, the idea is that first it stores some of the microbes, good microbes, like it's a pantry for depot for microbes. Okay, because when people get antibiotic therapy or something, the cecum can kind of grab from appendix and replenish the microbial populations. Another thing, appendix is surrounded by MALT, mucosa-associated lymphoid tissue, Peyer's patches, and so those Peyer's patches, we talked about mucosa-associated lymphoid tissues, the follicles that are in the mucosal lamina propria of the mucosal layer. There is a great likelihood that exposure of the Peyer's patches to certain microbes can shape your immune response. Sounds like Greek, right? Now, we live in a clean society. We do, right? I mean, we're pretty clean. We don't have like filth on the floor. Wash our hands. Okay, we wash our food. Okay, we don't forage food from the soil. Okay. Also, surprisingly, or not surprisingly, developed countries have highest rates of autoimmune diseases like allergies, Crohn's diseases. If you would go to third world countries, South America, Africa, oh yeah, they have a lot of parasites and diarrheal diseases and all that kind of infectious stuff, but they have pretty much no autoimmune diseases. So one of the ideas is that when the way they live, they pretty much eat dirt all the time. The kids eat dirt from the very first day, right? They don't wash hands very often. They expose the immune system through the Peyer's patches, through that mucosa-associated lymphoid tissue that's located in the large intestine. They expose the immune system to a wide variety of microbes. Okay, and that trains the immune system. Of course, not everybody gets through this training. Some folks die. But those who survive, the immune system is shaped properly, and they don't have autoimmune diseases. And there are observations that if you expose your digestive tract to, say, certain types of worms, that treats Crohn's disease. Okay, and it's now approved treatment for IBD, inflammatory bowel disease. They give you worms, not human worms, pig worms.
Like a fine line then between like exposing yourself to certain like pathogens and like stuff like that versus like.
Pretty much the idea that is now being actively promoted, the hygiene hypothesis, is that you know that joke about how mom treats pacifier with first, second, and third child? You know, with the third child, it's like this. Give it back. That's a good idea. Okay, let them lick the floor. You know, let them eat dirty foods. Are they going to have diarrhea? Probably so, regularly, but it will shape the immune system. Because if you clean it too much, the immune system, instead of being properly trained, quote unquote trained, will turn against the body and will start to attack the body tissues. We don't know the mechanism, we don't know why it happens, but the statistical data, the epidemiological data suggests more and more that, you know, we have to expose it. We have to stop being such a clean society. Like, take a shower once a week. That should be enough. Not that I'm suggesting. Smell awful, I suppose, but in terms of respiratory health and digestive health, it will be perfect. Okay. Colitis is one of the manifestations of Crohn's. When we say colitis, it's the inflammation of the colon. colitis does not necessarily have to be caused by immune responses, okay? Infections can cause it too. But one of the symptoms of Crohn's is colitis. Inflammation of the colon that may lead to ulcers, ulcerative colitis. And sometimes, the only way to treat it is to surgically remove the colon. Does that make sense? Well, no, wait. It's like, colitis is like cough. That makes sense? Cough or runny nose. Runny nose is not a disease, it's a symptom of infection. Colitis is the symptom of Crohn's disease. One of the symptoms. So colitis is inflammation. Ulcerative colitis is when you have ulcers, that when the epithelium is gone. And then ulcerative colitis may lead to the penetration of the intestinal wall and spillage. So it's not, so say inflammatory bowel disease is the umbrella term that includes Crohn's. Okay? Crohn's may affect the entire colon, it may affect parts of the small intestine. Okay? Colitis is specifically restricted to the colon. Does that make sense? Now, so now what we think the appendix functions, immune function and digestive in terms of the microbes. Okay? So now we don't just cut it out. So once the chyme passed that very, very important region, it goes to the ascending colon. Ascending and descending are both retroperitoneal, so they are behind the peritoneum. Okay. Transverse and sigmoid are surrounded by peritoneum, so they are intraperitoneal organs. Nothing too exciting is going on there. There's a little bit of the water absorption in the colon, but not a lot of excitement. Okay. So as chyme moves anatomically, goes through ascending to the hepatic flexure or right colic flexure. Hepatic because it's located close to the liver on the right side. Look at the dude right there. The next to that huge dude, the smaller dude with the liver exposed. Okay? You see the grayish colon part? Okay? And you see the transverse colon exposed? So to your left is the right colic flexure, where it bends. Where it bends right here. That's the right colic flexure. It's also called hepatic flexure because it's close to the liver. Okay? To your right is the left colic flexure or splenic flexure. Does that make sense? Okay, well, before we... Yeah. Yes, you should know both terms. And then gets to the sigmoid, okay? Oh, it's probably copy-paste. Sigmoid, and then retroperitoneal rectum. Does that make sense? It was my mistake here. There are no flexures here. Doesn't have a specific name. Does that make sense? Okay. Now, if you would look at the rectum, there are rectal valves in the rectum. The purpose of the rectal valve to separate two processes that rectum does: passing the gas and defecation. Okay? The presence of these valves actually allow humans to pass the gas without defecating. That's handy, yeah. There's a fine line, yeah, between the huge fart and a small shit. Yes. Say again?
I'm just saying, like, essentially, like, two different tubes.
No, it's one tube with the valve that prevents feces from going out but allows the gas to leave.
So maybe like a filter.
I wouldn't say it's a filter. Filters occupying, any filter is occupying an entire tube. It's flaps. Does that make sense? Like flaps. So they don't let feces to leave, but allow gas to leave. Yeah. Now, so all the way up until pretty much here to this line, pectinate line, the lining is simple columnar epithelium. After pectinate line, it's the stratified squamous. So here's the stratified squamous epithelium. Does that make sense? Because feces are hard. Like physically, they not, well, sometimes they are soft, sometimes they aren't. And they rub, they put abrasion force, right, against the rectum, against the anal orifice. So you have constant sloughing of the cells. Does that make sense? So that's why stratified squamous epithelium. Here you can see a couple more structures. So you have the columns, you know, the structures that kind of protrude. So anal columns, and between them anal sphincters. Essentially you have structure there like this. Okay, does that make sense? It's kind of wavy. The function of anal columns, no, doesn't make sense?
No, I'm just confused. Did you say sphincters?
It's not sphincters, anal sinuses. Thank you for correcting me, I misspoke. Anal sinuses, sorry. So anal columns and anal sinuses in between them. Okay? Like this. So these are two anal columns and this is anal sinus. They produce mucus to lubricate during defecation. Pressure, when feces approach the anal orifice, the pressure stimulates the mucus production. That makes sense? So they leave easier. Does that make sense? Now control of the movement is under two sphincters. The internal anal sphincter is involuntarily controlled by parasympathetic nervous system. Okay? The external anal sphincter is voluntarily controlled. Okay? So pretty much during defecation, we'll talk about it, but during defecation what happens when feces accumulate in the sigmoid colon and rectum, the stretch stimulates the relaxation of the internal anal sphincter. That makes sense? Then, and we are also aware of the urge for the defecation. Then we decide whether we go or not. So we consciously relax, or we keep external anal sphincter closed until the moment we defecate. Does that make sense? That's how it works.
I have a question
Sure.
regarding like the, I guess more so the colon, because we talked about like with digestion and stuff,
Mhm.
how soon after you start eating, that's kind of why you have to like use the restroom.
Mhm.
Basically, like if you were to eat a meal and then it's like you know, like the colon is like absorbing like the liquid and like whatever, but like does it kind of, I don't know how to word this, like why is it that all of a sudden like when we eat more then we have to go? Like is it like kind of like our fecal matter's just like hanging out in our colon?
Getting there. Not here. We're gonna talk about it here. Okay, just give me a couple. So few more anatomical moments. So this is you saw this already. Now the structures of the colon, you saw this one already, but the colonic structures are connected to each other by the number of serous membranes. Okay, so greater omentum. Now you can't really. So great on this picture of the cadaver, greater omentum is removed. Greater omentum is like an apron made of connective tissue and fat. And I have read some testimonials, let's put it this way, of medical students who said when they worked with cadavers who had obesity or who were overweight, they noticed that the intestines were covered, especially males, because of the visceral fat. It was a huge layer of fat just laying over the intestines. So, and that turned them into like healthy freaks. Now, other structures, so lesser omentum connects liver to the stomach and to the large intestine. Mesentery here recently was acknowledged as a separate organ. Yeah, so now it's an additional organ. That's pretty much what I have to tell you about this. That's an organ. Okay, a lot of lymphatic nodes here, there, mesenteric lymph nodes. Okay, so that's one of the function of mesentery is not only to keep stuff together, but also to provide, how to say, first stage of immune response. Because obviously, that's where the microbes can enter the body cavity. Does that make sense? And various mesocolons. The transverse, it connects transverse colon and parts of the small intestine. Mesentery, here, sigmoid mesocolon that attaches sigmoid colon to the small intestine and parts of the large one. Does that make sense? Now, this is the great illustration of what it means to be retroperitoneal and intraperitoneal. Okay, so you can see how the duodenum is retroperitoneal. Okay, does that make sense? Now, let's take a glance on what's going on. Why do we want to go to the restroom once we eat? First of all, microanatomy, the tissue structure, all the same, 4 layers: mucosa, submucosa, muscularis, and serosa. Okay, mucosal layer, which is most interesting for us, the columnar epithelium with the multiple goblet cells. Okay, that produce mucus. Mucus in this case serves mostly for the lubrication of what is becoming feces. Okay, absorptive cells in the large intestine are mostly responsible for the absorption of water, and yeah, that's pretty much it. Mostly water, right? But the immune structures, as I mentioned, the lymph nodes, the lymphoid follicles, they play a very important role in shaping the immune response. Does that make sense? It's mostly absorption and a lot of immune function. Now, if you would look at the structure of the large intestine, you would see several very fascinating anatomical features. One is these bags, sacs, that large intestine is divided into. Can you see them? It's fragmented, it's segmented. So this segment is called haustrum. Haustrum is singular form. Okay, plural is haustra. So you can see multiple haustra here. It allows you to they can contract separately. They can segment the chyme. That makes sense? So here, these edges, these boundaries of each individual haustrum, these boundaries can squeeze and separate the chyme into the kind of smaller pieces and smaller segments. So how is it important for segmentation? It is a smooth muscle that does it. Just the locations of the smooth muscle. So it's not, it doesn't do this. It does more like this. Okay? In segments. Right? So that's one, that's segmentation. That is a unique segmentation process that happens in the large intestine, so-called haustral churning. Okay? When they are more or less independently churn the, I want to say food, but it's not a food anymore. It's chyme and it's almost feces at this point. Does that make sense? That's one aspect of peristalsis. Another aspect is mass movements. That's what you were asking. So mass movements is when instead of slow, remember we talked about peristalsis in like small intestine, it's sluggish. It's slow to allow digestive juices to break down the chemicals or the nutrients. But during mass movements, the peristalsis becomes powerful and pretty fast. So this massive, powerful movements, powerful peristaltic movements, move the chyme / feces towards the rectum. Okay, so it propels them and accumulates them in the sigmoid colon. Okay, does that make sense? Okay, now that happens after the meal. You eat something and like 20-30 minutes you have an urge to go to the restroom. Can you tell me which hormone is responsible for stimulating mass movements in the large intestine? Okay, now the way you, let's learn how you think about such stuff. Do you know of any hormones that are produced in the mouth? I personally don't. If you know, let me know. I personally don't. So it's not a mouth. What is the next stop? Like important stop? Stomach. What is the hormone that is produced by the stomach when food is getting there? Gastrin. What gastrin does? Let's see what it does. Let's recall. What it does to stomach. Huh? Well, no, distension is the stimulus, but what it does, so contraction of the smooth muscles and, well, it's also a stimulus. Now, what gastrin stimulates in response to those changes? Huh? Secretion and peristalsis, right? Stomach secretion and stomach peristalsis. What gastrin will do to the small intestine? Yeah, it will increase the peristalsis. What will it do to the ileocecal valve? What do you think? Relax it. It will open it so the chyme can get from the small intestine into the large intestine. And finally, we need space in the large intestine now to accommodate incoming food. Mass movements will direct what is about to become feces towards sigmoid colon / rectum. Does that make sense? Now, this mass movements produced partially by this band of the smooth muscle. Let me get it over. So if you look at the structure of the colon here, you can see this band over here. Can you see it? This is called tinae coli. This band of the smooth muscles, when it contracts, it assists mass movements. Does that make sense? That's the function of tinae coli, to assist in mass movements. We're good? That more or less clear? We figured out the structures of the large intestine. Yes. Should be. Yeah, right here. Tinae coli.
So for like, because some people go like once every like 2 days, and then you go like 3 days.
Yeah.
So does that based on like
It's based on the diet, based on the microbes that you have. That's a great question, actually. When you switch diets, it often changes. You go to extremely like high fat, no carb diet, your peristalsis is going to become slower, especially if you have no fiber. Okay, so the bowel movements will become less frequent, and they will probably be more solid. Okay, microbes, like, yeah.
I didn't want to change the subject, but kind of same subject. Like, what about like timing? Like, certain people go like at the same time. Does that have to do with like
Circadian rhythms, absolutely. Circadian rhythms play a role, but think about this. We normally, if you think about your schedule, unless it's completely crazy schedule, usually we have breakfast at a certain time, we have lunch at around certain time, we have dinner at around certain time. So we eat at certain times. We eat at certain times because circadian rhythms also regulate appetite. They contribute to the appetite regulation. So we want to eat the morning, afternoon, and evening. Okay. Now, if you think about what's going on during like Christmas week or New Year's week, when your whole schedule is completely fucked up and you eat at most amazing times, like I had a couple of days ago, I had a proper meal at 2 a.m. like dinner. I already had dinner. It was second dinner. So when you live like this, your entire schedule, physiological schedule of digestion gets completely changed. That makes sense? Now, another interesting thing, what is considered to be sort of normal frequency of bowel movements in the United States? On average, I don't think CDC has the exact statistics. Once, twice a day, right? That's what we perceive as normal, right?
Isn't there something like the rule of threes, like no more than 3 times a day, but no less than
Something like that. Something like, so having one bowel movement in 2 days is still considered normal if it's regular, okay? Having 2 bowel movements a day is still normal. If you go to India, the average is 5 bowel movements a day. And that's normal. It has different structure though. It's softer, huh? Not only this, well, spices probably, microbes. They have completely different microbes. And if you would go there and live there long enough, you're also going to have 5 bowel movements a day. Okay? And they're going to have different consistency. Okay? There's actually a clinical term. I don't remember the definition verbatim. It's very strict, but pretty much when fecal matter accepts the shape of the vessel, it's called diarrhea. That's when it's diarrhea. When poop fills up the vessel, that's diarrhea. There's also a test with a stick. When you put the stick in, then lean it 45 degrees, and if stick remains, without moving, it's a solid fecal matter. If it starts to go down, then it's not solid. They're really clinical criteria to call something diarrhea. Now, we're going to talk about neural controls of peristalsis, why the heck there's a movement in the intestines, and then we're going to go for a break. So, smooth muscles in the intestines are connected to each other. They work together. It's the functional syncytium. That make sense? Right? So one cell gets stimulated by neurotransmitters from that varicosity, okay, and a bunch of other cells that are connected to it get stimulated as well. So they work all together by gap junctions, allow that communication between the smooth muscle cells. Right? Now it looks like there are cells, so-called cells of Cajal, that may serve as the pacemaker. So they produce waves of depolarization, so-called slow waves. Okay? So this graph shows you changes in the membrane potential over the time. So during the slow waves, cells, the smooth muscle cells become slightly depolarized, but not enough to cause the contraction. Okay? But at some point, they come closer to the threshold for action potential. And when there is an additional stimulus, like stretch or parasympathetic stimulation, cells go over the threshold for the action potential and they produce the action potential. Those spikes are action potentials. Does that make sense? And each action potential is is contraction. And that contraction is movement, essentially. Bless you. Does that make sense? Now, unlike in skeletal muscles, the depolarization is mediated by the slow channels. Remember when we talked about skeletal muscles channels, that depolarized skeletal muscles are fast channels. Here the channels are slow. Sodium and calcium flow into the cell slowly. Depolarization wave is slow, but it develops slower, but it also goes away slower. So the contraction is slow, but it lasts longer. Does that make sense? Good. So stretch, stimulation, or different sorts of vagal reflexes, the parasympathetic reflexes, or hormones can stimulate as well. Okay. Now, the reflex, so-called peristaltic reflexes specifically, responsible for the stretch-induced contractions. So how it works. This yellow thing is the food bolus that stretches out the intestinal wall. Are you keeping up with me? This are sensory receptors, sensory neurons. Keeping up? Sensory neurons perceive the stretch or chemical changes, okay? And they communicate that signal to the interneurons, the blue ones. Interneurons produce the response by stimulating motor neurons. But interesting thing is that upstream of the bolus, so closer to this end of the alimentary tract, closer to the beginning, motor neurons produce substance P or ACH. Those two chemicals stimulate the contraction of the smooth muscles. Downstream, motor neurons produce nitric oxide and vasoactive intestinal peptide, VIP. Let me do it. Vasoactive intestinal peptide. And nitric oxide and VIP stimulate relaxation. Does that make sense? So you see, there's a contraction up, relaxation on the bottom. Where would bolus go? Down. Does that make sense? That's your peristaltic reflex. Question? That's circular and longitudinal, both of them. Both of them, yes. Let's talk about defecation after the break. Okay.